Hey everyone, Jaime Sable here again. Earlier in the week, I told you about how we developed our synthetic biology cure at the University of Memphis. And this time I'm going to tell you in a little more detail how we made this work from what we did at the AR Cure workshop to then what we went home and actually put all the pieces together and developed our cure and got it running. So quickly want to also give a shout out once again to Dr. Judy Cole. She is absolutely my partner in this and is a major part of how this is moving forward. So when we went to AR Cure that first summer, we knew that we were interested in a lab experience. And we had a couple major goals. So we wanted to have authentic research experiences for students that weren't existing in our labs at the time. And we were really interested also in trying to support at-risk students. And so we know that students who transfer into our program don't tend to do as well in the program. And so we really wanted to target those students to get them involved in these kinds of experiences earlier in the program. But early in the program is also complicated. You know, our Bio 1 and Bio 2 are really large lecture courses, and so that's difficult to do in terms of the labs. We just have so many of them. So what we do have at Memphis, we have 5 core required courses that all of our majors take. So Bio 1 with lab, Bio 2 with lab, cell biology, genetics with a lab, and evolution. And so we really wanted to target one of these potentially at least so that we would have students who all of our majors were taking the course. So we really narrowed it down to cell biology and genetics. The pros for cell biology were for us that was the most obvious place to target these at-risk students. For most of our transfer students, if let's say they took Bio 1 and Bio 2 somewhere else and then they come in, typically cell biology is the first course they're taking with us. We also had more than one section of cell biology, but it is not as large as our Gen Bio 1 and Gen Bio 2. And so we knew that we would be able to look at these different sections to be able to have an idea of how students were doing in these when we added a cure lab. The class is really very eukaryote focused, and so we thought that it would also be helpful to do something bringing in prokaryotes to be able to round out that experience for them. The cons is that there was not a current lab section at all for cell biology, and getting the faculty to buy in, the cell biology faculty, was an issue. So if we were going to actually create a required lab as a part of cell biology, that was a potential issue. For genetics, we already had an existing lab there. The pro, again, we had multiple sections of that lab, so we could compare against what was existing versus what's happening if we make one of them a cure. The cons is that it is currently a classical genetics lab, and we knew the next piece is the faculty buy-in is necessary. The person who teaches that is very open to new ideas, but really does think that classical genetic lab is classical genetics, the fruit flies, is a really important part. And so we also knew that was potentially an issue in terms of making this a whole semester experience. The other thing is that genetics is pretty late in the program. So most of the time students are at least juniors, sometimes seniors before they take it. And so working with genetics really would have limited our ability to help out some of those students that were maybe struggling earlier in the program. So we had a couple other courses here that were not part of our core that we also considered. Both of these, though, again, they were only seniors that were taking them. And so that was, again, a reason we really didn't want to go that way. So, again, like I said, we were really interested in transfer success. Students who take the Gen Bio 1 and 2 sequence somewhere else tend to have more difficulty in our upper level classes. And so we really wanted to think about how can we help give them a leg up, basically, into our upper level classes. And so we had a couple things that were going on here. I've been a part of a group that was thinking about a Bridges to Baccalaureate grant that we think would really be a way to bridge going from particularly community colleges to the University of Memphis. And so we wanted this cure to be a part of that experience as well. And so we really started to think about this as cell biology being our best option. First course most transfer students take at U of M. And so then we were able to opened it up for anybody who was taking cell bio, but we targeted recruitment to transfer students. And so like I said before, we came up with this course. I'm not going to go into a lot of detail of what we did with the course because I did that in the other video. But this is what we settled on. So this was a co-rec with cell biology, not required, but we were then able to do some interesting research to look at what's happening with those students who are taking the cure as they're going through cell biology versus those who aren't. So again, a little bit of what our timeline looked like. We did AR Cure the summer of 2017. That fall, we did a lot of meetings. Judy and I met a lot to think about exactly how this was going to work. We started pulling things together. We started figuring out what equipment we already had and what we would need to get. And then we also did a couple of proposals to our college and our department, and we were able to get some money that way, which I'll talk about in just a minute. That spring, we did course approval paperwork. We were gathering all our supplies, and Nathan came for the seminar. That summer, we did a test run, and that's a really important part. I really encourage you to do that. We weren't getting cells to grow in the beginning, and all different kinds of things that you just don't even expect to be an issue, but it was. And so we needed to test reagents, and we needed to really have a sense of what was going on, make sure we had everything, troubleshoot all those things before students were getting started. So then we ran the course that first time in fall of 2018, and as I talked about before, we needed to make some changes, made those, and just ran it again this last fall. And we plan to run it every fall moving forward. So about the funding, we had a couple of sources that we were able to get. So we used a lot of existing things in the department, but at the University of Memphis, there's a technology access fee that is money that it's one of the student fees. And so every once in a while, they open it up for you to write a proposal to be able to get some money to basically bring in some more technology. And so we were able to get $3,000 through that, and that's how we bought all the things from Mini1, basically. And then we also were able to get some more money from our department. We got a TA spot, which was huge. That was a big deal. But also some money for some consumables. So I know Nathan talks a lot about the various ways for cost savings. Obviously, if you do have access to different ways to get funds, use those. But there are ways to still do this even if you don't get $6,000 from your university. So moving forward, what we're really thinking about is how these ongoing effects of the cures, how they affect students' engagement in research, student success in the major. I will say that we have had a number of our students who have already gone through the cure who have gone on to graduate school doing research or have taken jobs in research as technicians. And so that's a really exciting thing for us to see. All of the students who have taken our cures are still in the program, and that's also a great thing to see. We're also working on building some more cures. So we have the synthetic biology one, and we're working on maybe developing a cell biology one as well, and then a genomics and bioinformatics one. And so the idea here would be that students would have multiple exposures to cures, have a lot of different research experiences. And so that's where we're moving forward. So a few suggestions from me. Do a test run before the course starts. We did it in the summer before we ran it in the fall, and I'm so glad we did, because just little things that we didn't even think about since we hadn't run a course like this before. So do a test run through the whole process yourself. Make sure that your shaker stays at the right temperature. Ours wasn't. Things like that. Be flexible. Our schedule and our benchmarks changed during the semester. Once we got into it, it's that first time, once we got into it and realized that some of these things really just weren't going to be feasible for students to do. And so we did change that throughout the semester. Just make sure you're telling your students what's going on so that you get buy-in from them. The second time we did it, we did not have to change so much because we had done so much. But even still, there was some flexibility we needed to have. Don't reinvent the wheel. So use what works and reach out for help. So, even if you're not doing something exactly the same way that you saw it done at the workshop, that's okay. Do what works for you. And there are so many people that are really willing to help with all of this. Nathan has been a really great resource for us. But I've also had people reach out to me since I've got mine going to ask for help. And we're all really happy to give suggestions. So remember the questions you had and the problems you encountered at the workshop. This is maybe not exactly the same for you guys since you're not on the ground like we were doing these experiments, but that makes doing that test run even more important. But remember the mistakes you make, the challenges you have, because students will have the same problems and so many more. And so, I remember being somewhat frustrated at times that, a student asking the same question multiple times, but then remembering, oh, it didn't go that well for us when we did the test run. So, just remember that those kinds of things are just part of the game. Okay, so I didn't put my contact info here again, but you have it from the last video that I did. And again, I'm always happy to chat, to answer questions, to let you know how we did things. So good luck.